Chapter 9. Enzymes and Breakdown. Enzymes drive the chemical reduction of food into absorbable units, but their effectiveness depends on the structural state of the substrate they encounter. They do not initiate digestion in isolation. They act on material that has already been altered by mechanical breakdown and gastric acid. The intestine is equipped with a broad array of enzymes capable of cleaving proteins, fats, and carbohydrates, yet these enzymes require access to specific sites within each molecule. When the substrate arrives in a form that exposes those sites, breakdown proceeds with high efficiency. When it does not, enzyme activity becomes limited by physical constraints rather than by enzyme availability. Proteolytic enzymes in the small intestine complete the reduction of proteins into smaller peptides and amino acids. These enzymes are designed to act on substrates that have already been unfolded and partially cleaved in the stomach. When this preparation is adequate, the remaining peptide bonds are accessible and enzymatic action proceeds in a coordinated manner. When upstream unfolding is incomplete, large segments of protein remain structurally resistant. Enzymes engage only the exposed regions, leaving intact domains that pass further along the intestine. The result is incomplete reduction and reduced availability of absorbable amino acids. Lipolytic enzymes operate under a similar dependency. Fat digestion requires that lipids be dispersed into smaller units that can be accessed by enzymes. Bile plays a central role in this process by organizing fats into structures that increase surface area. When fats arrive in a properly prepared state, lipases can act efficiently, reducing triglycerides into components that can be absorbed. When fats remain in larger aggregates due to inadequate preparation, enzyme access is limited. Breakdown slows and portions of dietary fat continue through the intestine without being fully processed. Carbohydrate digestion also reflects this relationship between structure and enzymatic access. Enzymes act on specific linkages within carbohydrate molecules, and their activity depends on the availability of these linkages. When carbohydrates are presented in forms that allow enzyme contact, breakdown proceeds rapidly. When structural barriers persist, enzymatic action becomes uneven, producing a mixture of fully reduced and partially processed fragments. These fragments influence both absorption and microbial activity as they move through the intestine. Enzyme secretion is regulated by signals that reflect the presence and composition of incoming material. The pancreas adjusts its output based on what is detected in the intestine, increasing enzyme delivery when demand is higher. This regulation assumes that substrates are within a range that enzymes can effectively process. When substrates fall outside that range due to upstream deficits, increased enzyme output cannot fully compensate. The limitation remains structural, and the efficiency of breakdown is constrained regardless of enzyme concentration. The timing of enzyme activity is also critical. Enzymes must be present when substrates arrive in a state that allows interaction. When gastric emptying is irregular or when early phase signaling is weak, the alignment between enzyme release and substrate availability is disrupted. Enzymes may be released into the intestine before or after the optimal window for interaction, reducing overall efficiency. This misalignment contributes to variability in how completely different portions of a meal are processed. Enzymes and breakdown therefore function within the context of the entire digestive sequence. Their ability to reduce food into absorbable units depends on the preparation that occurs upstream and the timing of their release relative to substrate arrival. When the sequence is aligned, enzymes operate on substrates that are accessible and appropriately timed, producing consistent and efficient breakdown. When the sequence is disrupted, enzymatic activity is constrained by structural and temporal factors, and the products of digestion reflect that limitation. Pancreatic enzyme dependency on upstream signals. Pancreatic enzyme output is not constant. It is scaled and timed according to signals that originate before and during the transition from stomach to small intestine. These signals inform the pancreas about the volume and composition of the meal and determine how much enzyme is released and when that release occurs. The system assumes that upstream processes have prepared the substrate to a predictable state. When those signals are accurate and timely, enzyme delivery aligns with substrate arrival. When they are weak or mistimed, pancreatic output becomes mismatched to the material entering the intestine. Early signaling begins before food reaches the small intestine. Sensory input and gastric activity generate neural signals that prime pancreatic secretion. This preparatory phase raises baseline output so that enzymes are available as soon as partially processed material begins to enter the duodenum. As gastric contents move forward, additional signals are generated based on the chemical composition of the incoming material. These signals further adjust enzyme release, increasing output when proteins, fats, or carbohydrates are present in forms that require breakdown. The effectiveness of this system depends on the integrity of upstream processing. When proteins have been unfolded and partially cleaved in the stomach, and when fats have been exposed and dispersed, the signals generated at the intestinal level accurately reflect the state of the substrate. The pancreas responds by delivering enzymes that can immediately act on accessible targets. Breakdown proceeds with coordination between substrate and enzyme, and the sequence advances efficiently. When upstream processing is incomplete, the signals that drive pancreatic output become less reliable. Larger, less processed substrates may not trigger the same level of response, or they may generate signals that do not correspond to the actual difficulty of breakdown. The pancreas may release enzymes in amounts that are insufficient relative to the structural resistance of the material, or the timing of release may not align with substrate arrival. In these conditions, enzymes are present but cannot fully engage with the substrate, and the efficiency of digestion declines. Timing is a critical component of this dependency. Enzymes must be delivered into the intestine at the moment when substrates are available and accessible. When gastric emptying is irregular or when early phase signaling is reduced, this alignment is disrupted. Enzymes may be released too early before substrates arrive, or too late after portions of the substrate have already moved beyond the optimal region for interaction. This temporal mismatch reduces the effectiveness of enzymatic breakdown even when total enzyme output is adequate. The system attempts to compensate for these mismatches by adjusting enzyme secretion based on ongoing feedback, but this compensation is limited by the structural state of the substrate. Increasing enzyme concentration does not overcome restricted access to cleavage sites within intact or poorly prepared molecules. The limitation remains at the level of substrate preparation, and the pancreas continues to operate within constraints that it cannot resolve independently. Pancreatic enzyme dependency on upstream signals therefore reflects the integrated nature of digestion. The pancreas relies on accurate and timely information from earlier stages to align its output with the needs of the system. When upstream processes are functioning properly, this alignment allows enzymes to operate at full efficiency. When they are not, pancreatic output becomes misaligned with substrate conditions, and the breakdown of food proceeds with reduced consistency and effectiveness. Protein, fat, and carbohydrate processing. The breakdown of proteins, fats, and carbohydrates follows a coordinated sequence in which each macronutrient is transformed according to its structure and the conditions established upstream. These processes do not operate independently. They are linked through shared requirements for timing, substrate preparation, and signaling. The efficiency of each pathway depends on whether the material entering the small intestine has been properly altered in the stomach and whether enzyme delivery and bile coordination align with that material. Protein processing begins with acid-driven unfolding in the stomach, which exposes peptide bonds and allows proteolytic enzymes to initiate cleavage. By the time proteins reach the small intestine, they are expected to be in the form of smaller peptides. Pancreatic enzymes then complete the reduction into amino acids and short peptide fragments that can be transported across the intestinal lining. When this sequence is aligned, protein breakdown proceeds with high efficiency because enzymes are acting on substrates that are structurally accessible. When unfolding and early cleavage are incomplete, larger protein fragments persist, limiting enzymatic access and reducing the availability of absorbable amino acids. Fat processing depends on both structural dispersion and coordinated bile delivery. Lipids must be broken into smaller units that can be acted on by lipolytic enzymes. Bile organizes fats into structures that increase surface area, allowing enzymes to access and cleave them into absorbable components. This process requires that fats arrive in a state that can be effectively emulsified. When upstream preparation exposes lipids adequately and signaling aligns bile release with substrate arrival, breakdown proceeds efficiently. When fats remain in larger aggregates or bile delivery is mistimed, enzymatic access is reduced and portions of dietary fat move through the intestine without being fully processed. Carbohydrate processing involves enzymatic cleavage of specific linkages within polysaccharides and disaccharides. These enzymes act on substrates that are generally more accessible than proteins and fats, but their efficiency still depends on proper timing and interaction with the intestinal surface. When carbohydrates are presented in forms that allow consistent enzyme contact, breakdown into simple sugars occurs rapidly and absorption follows. When structural barriers persist or when timing is misaligned, enzymatic action becomes uneven, producing a mixture of fully reduced and partially processed fragments that influence both absorption and microbial activity. The coordination between these pathways is essential because they share the same environment within the intestine. Enzyme secretion, bile delivery, and motility patterns must accommodate all three macronutrients simultaneously. When upstream signaling is accurate, the system adjusts to the composition of the meal, scaling output and timing to match the demands of each component. When signaling is weak or when substrate preparation is incomplete, this coordination breaks down. One pathway may dominate or require compensation, altering the conditions under which the others operate. Processing efficiency therefore depends on the alignment of structure, timing, and signaling across all macronutrients. Proteins require unfolding and cleavage, fats require dispersion and emulsification, and carbohydrates require accessible linkages and consistent enzyme interaction. Each pathway builds on the work of the previous stage, and disruptions in one area can influence the others through shared mechanisms of motility, secretion, and substrate availability. When the sequence is aligned, all three pathways operate in concert, producing substrates that can be absorbed with minimal variability. When it is not, each pathway reflects the constraints imposed by upstream failure, and the overall efficiency of digestion declines.